Well, it is an honor to introduce Dr. Bruce Beutler, a leading scientist in modern immunology whose work has advanced our knowledge of innate immune system. Dr. Beutler has gained international recognition for the discovery of toll-like receptor 4 and demonstrated that it detects bacterial LPS. This discovery established a fundamental structure for understanding how the innate immune system senses microbial products and initiates inflammatory signaling. As you probably know, the Nobel Prize in Physiology and Medicine was awarded to him in 2011 in recognition of these contributions. He currently serves as professor and director of the Center for Genetics of Host Defense at the University of Texas Southwestern Medical Center, where he leads research to identify genetic determinants of host defense and inflammatory responses. We are truly privileged to have him join us today, so let us give a warm welcome to Dr. Beutler, please.
Thank you all very much. It's really a pleasure to be here. I ask you a little bit of patience so I can get my talk on the screen. Okay, very good. Thank you very much. It's really a pleasure to be here. I've been to Mexico several times, but never to Mexico City and never to Monterrey, and those are the two places I've visited on this trip, and it makes me want to come back very often. I was actually at one of your meetings, I don't remember how many years ago it was, maybe about 25 years ago, and I don't remember where it was. Maybe, were you ever... It was Ixtapas, Zihuatanejo. Yes, I remember. That was what I was about to guess, and I'm glad you told me. But I thought then, as I do now, that some of the most scholarly immunologists are in Mexico. I know that there's a shortage of resources sometimes to do experimental work, but bright people find other things to do, notably to think about biological processes. And I had some very interesting conversations back then, which I haven't forgotten to this day. I'm going to talk about something very different than I did 25 years ago, and that's what I think is the most exciting thing going on in my laboratory these days. As the title says, we use forward genetics to find new targets for cancer therapy, and I'll tell you how that works, but I will say that most of the credit for this work goes to Hsin Shi, who did a screen for cancer cell extrinsic mutations that cause resistance to cancer, and Jin Choi, who did a cancer intrinsic screen, and I'll explain how that works. I also want to tell you that I am the co-founder of a small company called Potentium Biosciences that tries to translate the discoveries we have made. And for that reason, unfortunately, I can't be completely transparent about what some of the targets are, but I will discuss some of them. There's a background to the story I'll tell you today. It didn't come from nowhere. Early in my career, I learned to work with proteins, how to isolate them, how to separate them, and I became very good at that. And at some point, I did a postdoctoral fellowship after I received my MD degree. I wanted to work on something that was medically relevant. I was set to work trying to isolate a factor called cachectin, which was supposed to be responsible for wasting in chronic disease. This was an LPS-induced macrophage factor, which I took account of, and I isolated the cachectin activity using an assay that measured the shutoff of lipoprotein lipase in fat cells, preventing them from taking up exogenous lipid. I found that the factor we called cachectin was the mouse ortholog of tumor necrosis factor, which was identified contemporaneously by workers at Genentech. And I felt that it was a major mediator of LPS-induced shock. I demonstrated that by passively immunizing mice against TNF and showing that they were somewhat resistant to LPS. And thereafter, together with a graduate student and a postdoc working in my lab in Dallas, I invented inhibitors of TNF, which we patented and which we sold to a company called Immunex. And to this day, as Enbrel, these inhibitors are still used to treat inflammatory diseases. On the basic side, I was mostly interested in how LPS would activate macrophages in the first place. And I was very much aware that there was no receptor for LPS known. I tried to use my biochemical skills to purify the LPS receptor, and I got nowhere. I also used expression cDNA cloning, and that got me nowhere also. And eventually, I began to use classical genetics. I made use of 2 strains of mice that were unrelated to each other, each of which had an allelic mutation that had been mapped to chromosome 4 by other workers that completely prevented the response to LPS. It was a qualitative mutation, a qualitative phenotype. Either mice would respond fully or they would not, and it was a recessive mutation in each case. And there was a failure of the 2 strains to complement when mated. So I thought this was very likely the LPS receptor, specific as it was, and I used positional cloning to track it down. This took a total of 5 years between 1993 and 1998. And it was a kind of a nerve-wracking process because I was aware that there were several groups trying to positionally clone the LPS locus. We finally found it just all at once. It was the only authentic gene in a huge contig of bacterial artificial chromosomes that spanned about 5.8 million base pairs on mouse chromosome 4, and we had sequenced about 90% of it before we encountered the relevant gene and found, in one case, a point mutation, in the other case, deletion of the gene, and that gene was toll-like receptor 4, and this was the first really strong evidence that this protein had evolved to detect molecules of microbial origin, specifically LPS in this case, and probably nothing else. And later it was found that there were many toll-like receptors, 10 in humans, and 12 in mice, and each recognized a different sort of signature molecule that would activate the innate immune response. Even though it had been nerve-wracking, as I said, I loved the approach of going from a phenotype to finding a discrete mutation and knowing for sure that there was cause and effect without any equivocation at all. I wanted to do more of it, to study other aspects of innate immunity. However, there were very few off-the-shelf mutations that had any relevance to innate immunity. For that reason, I decided I would make new phenotypes, which I could work on alone. And the way to make phenotypes in mice is with a germline mutagen called ethyl nitrosourea, or ENU. And one simply injects mice of a certain background, in our case C57 black 6, with ENU, then breeds them to black 6 females, and one gets male mice that are founders of large pedigrees, or small ones, as we practiced in the beginning. And these mice are recessive for mutations that were induced in the spermatogonia of the G0. Each such mouse was crossed to a black 6 female in the beginning, and then with about 2 daughters, one would back cross to the G1 to produce a 3rd generation where there could be homozygosity for these mutations. At the time, we didn't know how many mutations were present in each G1 mouse. We made about 200 phenotypes during the years I was at Scripps Research Institute between 2000 and 2011, and we tracked down almost all of them, and we found that every single one that we identified caused a change in coding sense or in splicing function. Essentially, only qualitative phenotypes could be solved in those days. If you had much overlap between the phenotype of the heterozygous population and homozygotes, it was very hard going. These are some examples of visible phenotypes that we found just for show, but we were mainly interested in immunologic phenotypes. I stressed that only the causative mutation was found in those days. We would map to a small interval, and usually it contained just one mutation, and we ignored all the others that there might be. Tracking down a phenotype to the mutation, even in the best of times, took about 2 years, and that was true even after massively parallel sequencing methods arose, because you still had to rule out all the mutations there were that you had induced. I moved back to UT Southwestern in 2011, and having some greater credibility, I guess, from getting the Nobel Prize, I had a new startup package, and I had an idea about how to shorten the time for finding the cause of a phenotype. I wanted to make an automated system for meiotic mapping, and I wanted to do it in no time at all. And that is a reality now. These days, when we detect a phenotype, we know its cause at the same moment. Now, how is that possible? First of all, we proactively sequence the whole exome of every G1 mouse as soon as the G1 mouse is weaned. And these mice are then crossed to make very large pedigrees, not just small ones as in the old days, which was done for the sake of thrift. We typically make 10 to 15 G2 daughters from a G1 mouse and backcross them all systematically to the founder to get about 40 or 50 G3 animals. These G3 animals are then genotyped at every mutation site that was detected in the G1 animal. All of the coding and splicing changes there can be, essentially. And we know now that there are an average of 60 mutations per G1 based on thousands and thousands of G1 mice that we've sequenced. At the end of that process, one knows the zygosity of every mutation in the G1, whether it's homozygous, heterozygous, or reference allele homozygous in every one of these G3 mice. And all of the G3 mice are labeled, and they are subjected then to phenotypic screening. They are put into a pipeline, and about 400 mice per week are put into that pipeline, which might mean something like 10 pedigrees every week. And the mice are examined in quantitative screens for the most part, although some qualitative mutations are picked up and not ignored. And these screens go from very benign, non-invasive things like weighing the animal or testing its motor abilities in various ways, to taking macrophages and doing innate immune screens, immunizing them in multiple ways, doing FACS analysis, measuring antibodies, measuring also allergic type responses in the mice. We X-ray them and do DEXA scanning to look at bone density and size and morphology. We look at blood pressure, heart rate. We look also at the effects of DSS, a cytotoxin that can kill the epithelial cells of the bowel, and try to find exaggerated or resistant phenotypes. And then the pipeline usually is split up into looking at other things. And these are done partly in our lab, partly in other labs at UT Southwestern. Some mutations are only findable if you start with a fresh set that hasn't been tampered with at all. An autism screen, for example, that looks for mice with unusual autism-like characteristics. And believe it or not, there are humans that have orthologous mutations that have classical kinds of autism. When the phenotypic data are uploaded from any screen for a complete pedigree, the computer is automatically triggered to do a statistical computation. And that's what you see here. For every mutation represented by a dot and shown from spatially from chromosome 1 to chromosome 19 or 20 in some cases, we look to ask the question, does the distribution of phenotypes and genotypes reject the null hypothesis that this mutation has nothing to do with the phenotype that was observed? Now you see the pink line, that's the p.05 level of confidence. The red line represents p.05 with Bonferroni correction for the large number of mutations we're looking at in that pedigree. And you see that there was really robust rejection of the null hypothesis for one of the mutations, in this case on chromosome 1, with a p-value of better than 1 in a billion. And if you were to click on that point, you'd immediately be referred to the data, and Here you see the phenotypic performance of homozygotes, which is notably different than heterozygotes, reference allele mice, or wild-type mice that have no mutations at all. So irrelevant data are never brought to our attention, but if there is some excursion from normal among the mice, and if it has a genetic cause, that's brought to our attention. And this way we find mutations almost in every pedigree we look at. Here's another example. We have this kind of skewed distribution of phenotypic performance in homozygotes, and we have some hitchhiker effect where numerous mutations on chromosome 17 are carried along, but one of them clearly has the best linkage. Usually that's the causative mutation, though not absolutely always. And again, this would be an outstanding candidate. It's not always so clear-cut as that. Sometimes mutations co-segregate. Sometimes a screen doesn't work for a particular mouse, and you have too few phenotypic assays, and things that are in linkage are showing exaggerated concordance with the phenotype compared to the really causative mutation. And that's an aberration, but we're aware of those sorts of things happening. And when there's any confusion at all looking at what the causative mutation is, we did in the beginning make a human algorithm where we considered every factor that we thought we should consider in deciding what was really causative. And it did for us humans just a little bit better than nothing at all, than just using the statistics. And that is to say, when we looked at a large number of pedigrees, and we recreated the mutation in isolation and then asked in an expanded CRISPR pedigree, is the mutation really causative of what we saw or is it not? Sometimes it was, sometimes not. And as a diagnostic, the human algorithm gave something like 60% area under the curve. At that point, back in about 2015, before AI was the huge thing it is now, we decided we would make a real machine learning program, and we took all of these factors, plus some embedded machine learning programs that we had made looking at protein damage or gene essentiality, and these 64 human factors as well. We made a program called Candidate Explorer, and it would give a score of 0 to 1 that we felt might be better than what we humans could do, and indeed it was. It gave an excellent diagnostic of the causative mutation with an area under the curve of nearly 0.95%. Again, looking at 5,888 phenotype genotype correlations where we had recreated the mutation or knocked out the gene or both. And that was the training set that we used. And as you see, this Candidate Explorer score has a curvilinear relationship to the probability of verification by CRISPR. Now that looks interesting, but I want to show you the same data in a different format. On this training set, the Candidate Explorer program works so that the frequency of mutations that fall in the mid-range is diminished. We're reducing the uncertainty in general, and mutations cluster either as being very probably true or very probably untrue. And we have enough mutations of the many thousands that we create every year that we can focus on this part of the curve. These things we know are probably true, and so only those we move forward with if they're not in the training set but in the real world. And we make a CRISPR allele and we verify it, and we then, if it's phenotypically interesting to us, we go forward and we do a lot of other studies to understand mechanism. Using this system as well as the old one, we did solve a lot of questions in innate immunity. Here every box represents a different gene, and sometimes you see there are many alleles that we identified, and we found where TLR signaling was concerned a number of new things. We found genes encoding proteins that were needed to escort TLRs to where they needed to be in order to signal, that would condition the endosome. We found that Hermansky-Pudlak syndrome mutations affected TLR signaling. There were new genes that were involved in the processing of TNF that we identified, or its transport to the surface. And we found new adapter proteins and so forth. So all that was very interesting. Eventually, as of the last time I looked at mutagenetics, our database, we had 30,895 instances of phenotypes where we could say this phenotype is caused by this mutation with a very high level of confidence. And those cases emanated from 5,895 mutations that resided in 2,559 genes. That was kind of the breadth of genes that were disclosed by the phenotypic screens that we were using. We could have used more, I suppose, but we are only human. And in many cases, we made new diseases, which in some cases subsequently were verified with orthologous human diseases. But of greater interest with time, we began to think that modifying existing diseases, complex genetic diseases in the beginning, and then sometimes environmentally induced disease or monogenic disease, could be more interesting than creating a new disease. Particularly if people didn't understand the disease process very well, modifying the disease with a single mutation can tell you something about pathogenesis. Sometimes we could exacerbate a disease or accelerate it or augment it. Sometimes we could strongly suppress a disease. The example we started with was an autoimmune disease, type 1 diabetes, as modeled in the NOD mouse. And here we worked with Lucien Chateauneud and Jean-François Bach at the Necker Hospital in Paris, and they had maintained a very high incidence strain called NOD-NCKH. And looking in a pilot study with only 14 pedigrees, we found 7 instances of accelerated type 1 diabetes, as shown here. You have 8 mice here, and we're looking at the time to glycosuria, homozygotes for a certain mutation, in this case in an aminopeptidase called XPNPEP1, get precocious diabetes. And on the other hand, we found mutations like one in coronin 1A that made the mice never get diabetes. Now that was less interesting than you might think, because it's well known that coronin 1A in humans and in mice makes a kind of skid syndrome, and these animals didn't generate enough T cells or B cells to support an autoimmune response, and I regard that simply as proof of concept. But we've gone further with this, and we have some other mutations that are interesting, really quite novel, that don't 100% block type 1 diabetes, but certainly have a very strong inhibitory impact on it. If you have such a mutation, of course, you can begin to think, might it be a good target that we've discovered for some kind of therapy or preventive measure? Now, it's probably known to you that there are certain suppressor alleles that have been found in people that confer disease resistance, but very few of them, surprisingly few. One that comes to mind is maraviroc. This was a drug that was developed when it was discovered that a mutation quite prevalent in Northern European populations was in CCR5 and truncated the molecule, and that caused people with HIV infection to be very resistant to progression to AIDS. And this elite resistance was caused because there wasn't fusion of the virion with CD4 T cells. The drug Repatha was found in a similar way. GWAS studies revealed that people with mutation in PCSK9, mostly people in West Africa, had an increased LDL uptake of cholesterol, and therefore cholesterol synthesis was suppressed, and this can be used instead of statins. A blocker of PCSK9 action is used as an alternative to statins. And there are a few more things that one might think of. The drugs of the class, the Jardians as a representative, were found because the mechanism of tubular reclamation of glucose in diabetes is upset by a channel mutation. Jardians blocks that channel. People with type 2 diabetes maintain a low blood glucose simply by passing glucose in their urine until it reaches a low physiologic level. But there are very few of them, And I wanted to ask whether we could challenge something that is really robust, a very serious disease. Could we suppress cancer? And in humans, if this happened, you have to think that it would be difficult to know about it. It might be that one could address it with GWAS, but if you have someone without cancer, that would be like 3/4 of us that never develop cancer in the course of our lives, but die of something else. And that means that you have a limited opportunity to find something new. Really, you might have many such loci. It would be hard to do family studies. The only way to do it that I can think of is to start with a family that has something like Li-Fraumeni syndrome, and you look in an extended pedigree to see if you can find a mutation that correlates with the lack of disease. But very difficult to do, can be addressed much more incisively with forward genetics in mice, where you're looking at a handful of mutations in every pedigree, and you have the power to say this is causative or this is not. If you think about it, resistance could operate in a cancer cell extrinsic way, which might mean immune control of the growth of the cancer, or it could be in a cancer cell intrinsic manner, and different screens can address each of those possibilities. To begin with, I'll talk about He-Sin Shi's screen for extrinsic resistance to cancer. He used 2 transplantable syngeneic cancers, B16F10 or MC38, and while these are somewhat heterogeneous cell populations that one administers, they're constant. We're only mutagenizing the mouse genome. We're giving G3 mice from many different pedigrees an inoculation that's normally lethal to mice that have to be sacked within about 20 days. For B16F10, we sensitized the screen by giving anti-PD-1 antibody, knowing that by itself in a wild-type mouse, anti-PD-1 had no protective effect at all against that cancer. For the MC38 screen, we didn't give anti-PD-1 because it's curative of cancer. On days 13 and 20, the volume of the tumor was measured, and if there was no tumor, we did a repeat screen on the same pedigree, the same mice, to be sure that we hadn't made some kind of mistake. Using this screen, we found, first of all, a number of mutations that gave absolute resistance to B16F10 or MC38, sometimes both cancers. And these were very exciting at first, but there was a trivial explanation. In every case shown here in item 1, we have modified a class 1 presented peptide by a single amino acid. A missense mutation has occurred. And that leads to a situation where there is no longer negative selection for the wild-type peptide. And for that reason, the mice develop non-eliminated clones of T-cell receptors, of T-cells, that react strongly against the incoming tumor, which has the wild-type form of the peptide. And that's sufficient, that single amino acid difference in each of these cases, to cause robust rejection of the cancer. I held out hopes that this might still be a therapeutic route for us, but when I made TCR clones that would react with the peptide and made retrogenic mice and could give them to wild-type animals, in those cases, very severe autoimmune phenotype developed. It was not anything we could work with. On the other hand, there were some mutations that had a more general effect on immune function, and I'll tell you about 2 of them, citation and oddball, which work by very similar mechanisms, so they have different targets. Citation was first seen in our screen, as you see it here. It was just barely significant above the Bonferroni correction limit, and that speaks to the fact that this screen is not very good. The cancers grow at quite different rates in different mice in the populations, but you can see that with homozygosity for this mutation, the growth of the cancer is very much suppressed in all of the mice, and that made us think that this was probably causative, and Candidate Explorer agreed. The mutation, the citation allele, is a null allele of H2AA, which is something that codes for the alpha subunit of MHC class II, the only MHC class II complex in C57 black 6 mice. The protein is simply not expressed at all. It's a splicing problem, and you can see that there's practically no class II expression in homozygotes for citation as compared with wild-type homozygous mice. The mutation has a heterozygote effect once we expanded large populations of those mice, and you get really elite resistance to B16F10 melanoma, but significant resistance to other melanomas as well, like the YUM1 melanoma, and you have also inhibition of MC38 growth in the absence of a checkpoint inhibitor. To look to see whether there was cooperativity with anti-PD-1, we had to use a slightly different assay of cancer growth, intravenous administration of B16F10 cells, because there was such strong suppression in the case of subcutaneous cancers. If we looked with wild-type mice, either with or without PD-1 antibody given, those are the black points, you see really no difference at all, just as I told you. You have a slight protective effect of citation by itself, but if you give anti-PD-1, you have a great increase in survival time, at least, though all the mice ultimately die. And that makes sense in the context of what I'll tell you about the mechanism. We did many experiments, including making mixed chimeric mice and also doing conditional knockouts to see what population of cells supported the phenotype when you had this mutation. And the answer turned out to be conventional dendritic cells, and specifically we had a very robust FACS phenotype where CDC1 cells were suppressed in numbers in the spleen of citation homozygotes, and there was an excess of CDC2 dendritic cells. CDC1 had long been regarded as the population that's mainly involved in cross-priming. When you have a cancer, you acquire antigen in dendritic cells, and those are presented to CD8 T cells to activate a cytolytic response. And this was therefore a bit curious to us at first. If we looked at splenic CDC2 cells, we found that they were more abundant in total numbers. Also, they had a higher representation of class 1 MHC, even though they lacked class 2. And there was also a higher level of co-stimulatory proteins like CD80. We did then another critical experiment where we took B16F10 ova tumors to use ova as a target antigen. And these tumors were grown in mice, and we then harvested and enriched CDC1 or CDC2 cells from draining lymph nodes. We co-cultured those cells together with OT1 cells from OT1 mouse splenocytes, and we looked at the priming effect of CDC1 cells or CDC2 cells. With CDC1 cells, looking at citation in blue and at wild type in red, we saw no difference in the priming effect. And I want to point out that the priming effect is weak. That's all you can expect for a priming effect in wild-type mice, which explains why these mice can't really control the growth of the tumor. On the other hand, in CDC2 cells, while wild-type CDC2s gave no priming at all, true to what people know about them, the citation CDC2 cells gave really robust priming as compared to CDC1. And it's this priming effect that explains the elite control of the growth of cancer cells, all the more so if you add a checkpoint inhibitor. CDC2 cells now have a really new phenotype. They're a new kind of cell in the homozygous mutant mice than they are in the wild type. We did a lot of gene expression studies, and we found that, generally speaking, those proteins involved in the transport and processing and presentation of class 1 peptides were increased in expression in CDC2 cells, in citation homozygotes compared to wild type. And on the other hand, things that downregulated the response were decreased in expression. And the mechanism for it still remains to be nailed down. How is it that lack of class II has all these effects on dendritic cells? Well, first of all, I can tell you that if you grow wild type and citation dendritic cells in mixed chimeric mice, the effect is dendritic cell intrinsic. You can look at these two populations separately, and you see the phenotype in CDC1 and CDC2 cells from the mutants, and the wild type are just as wild type normally are. This doesn't mean, however, that there isn't another cellular factor cooperating with those dendritic cells. And it's my impression that that probably is the case. In fact, it's our overall hypothesis, yet to be fully verified, that one can disconnect the CDC2 cells from T regulatory cells, and that's what leads to the acquisition of this new hyperpriming phenotype. But we wanted to know how acutely the shortage of class II antigen would work. Therefore, we made conditional alleles of H2AA, an allele that we could induce knockout of with tamoxifen administration. And we found if we gave tamoxifen 5, 6, and 7 days after the tumor had been growing in such a mouse, that would lead to the phenotype, as compared to the case where we didn't give tamoxifen. That's the main comparator to use. We also had another almost perfect phenocopy of the original citation mutation, which we called oddball. And that mutation we saw right away was in C2TA, which is the master regulator of expression of class II chains in mice as in humans. And in this case, we again got elite resistance to the growth of B16F10. And again, we made conditional knockouts. We did the same treatment with tamoxifen on days 5, 6, and 7, and we found that the oddball allele, which is shown in red, would overlap perfectly with the floxed allele to which tamoxifen had been given in blue. So again, you get a very quick response, a quick change in the phenotype leading to priming. What could we use as a therapeutic, a rough and ready therapeutic? We could use antibodies. So we made monoclonal antibodies that would prevent class II mediated priming in vitro. And these antibodies were grown in fairly large quantity and given repeatedly to mice over a time course, and in a dose-dependent manner, they would block the growth of the melanoma, not by any effect on the melanoma, which didn't have any class II expression at all, but by an effect on dendritic cells. We found that this antibody, shown here in green, you can see antibody plus anti-PD-1, as opposed to antibody alone. It would work better if anti-PD-1 antibody was given as well. Interestingly, the antibody treatment does something a little bit unexpected as compared to the mutation, which has been present in the mice all of their lives, including in their developmental life. You see that antibody administration induces a new kind of dendritic cell, phenotypically using XCR1 and SERPA markers. And we call these X-CDC1 cells, thinking that they're probably derived from the original CDC1 pool, although that also is not 100% clear. These cells have a very striking phenotype. They're even better than CDC2 cells in terms of their augmented expression of class I and also their augmented expression of CD80. And they're recoverable as TILs from tumors and from draining lymph nodes after antibody treatment. All of this works by causing priming of CD8 T cells to targets that aren't normally recognized. We can look at that by doing single-cell sequencing and showing in naive mice that don't have tumor inoculation that you have a huge range of TCRs, usually seen only one time out of thousands. After you give a tumor to a wild-type mouse, you do see expansion of clones within the tumor itself among TILs. But de facto, these aren't really doing very much, at least in a therapeutic sense, because the tumor grows and grows, and you do no better in these wild-type mice than you do in a RAG knockout mouse, for example. On the other hand, you see unique specificities that expand, and these are sort of public specificities in the mutant, and we can demonstrate that these do have a therapeutic effect by making retrogenic mice that only have those individual clones in large numbers and challenging them with tumors. So here you see an example of the wild-type clone, or maybe a mixture of clones in this case, in a RAG recipient, and they have no therapeutic effect over RAG alone. But those colored clones that I showed you a moment ago, they do restrict the growth of the tumor in varying degrees. And the question is, what do they recognize? And we have some ideas about this, and we think it might be a kind of diagnostic to give a human patient an antibody for a week or two and see just what does expand and what might be therapeutic to treatment there forward. Now, I want to stress that these mutations work in a cancer cell extrinsic manner, and most of those things that we have found operate clearly by immune mechanisms. But we were interested to see whether we could find in a cancer-prone strain resistance mutations that operated within a cancer cell intrinsic manner. And to do this, we had to take quite a different screening approach. What I will show you are results with a tamoxifen-inducible KPC mouse model of pancreatic cancer. In this model, the P refers to PDX1, which is a driver of Cre in pancreatic ductal and acinar cells. And the Cre is a hybrid molecule with ERT2, the estrogen receptor, so it's tamoxifen responsive and goes to the nucleus, and the Cre activity can be manifested there, and floxed cassettes can be knocked out. In the cells, we also have a G12D KRAS mutation with a lock-stop-locks cassette, so it's not normally expressed, and there's no cancer until you give tamoxifen. P53 is also floxed in the homozygous state, so P53 knockout occurs in the pancreatic cells when tamoxifen is given. And one gets 100% penetrant pancreatic cancer that's lethal within a few months after induction with tamoxifen. And the question is, can we find ENU mutations that suppress this? Here is an example of one, which we call MUT1, and this is something we've studied a lot, and it has absolutely a stunning effect on transformation of pancreatic acinar cells and the process known as acinar ductal metaplasia. But here you see simply the statistics. In the red curve, we're looking at mice that have the knockout, that have a wild-type homozygous allele of MUT1. And on the other hand, we have no Cre driver in the case of the orange cells. And in the case of the green, we have a floxed MUT1 gene, which is knocked out at the same time that KRAS is activated and P53 is knocked out. And there's hardly any death there. And in the one mouse that died, it didn't die of pancreatic cancer. It died of something else. I don't really know what. But the pancreas, as you'll see, remains normal. Now, how normal? Well, here, without Cre driver again, you see that control. And you see a typical looking pancreas with lots of acinar cells, some ducts, occasional tangentially cut islets. All of you who've taken histology would call that a normal pancreas. If you look at the pancreas of the mouse that has been induced with a wild-type MUT1 allele, so it does get pancreatic cancer, you see no normal pancreatic tissue at all. Practically every acinar cell has undergone ADM, and you have a mass of ductal tissue and a lot of anaplastic cells. And if you looked further, as I did in this mouse, you see that there are lots of foci of metastasis in the liver and some in the spleen as well. And this mouse was nearly dead when it was sacrificed. If, on the other hand, you knock out the MUT1 gene at the same time you're activating KRAS and knocking out P53, you again see a normal profile in the pancreas. You see normal acinar cells, no foci of malignancy, and these mice, as I showed you, live on and are really quite normal in all their behavior. So I was astonished by that. I'd never seen such inhibition of ADM, nor had anyone else, and I told Jin Choi that I hadn't seen anything so amazing in several years, and we discussed immediately what we had to do with it. And the question was, well, okay, maybe it's just very good at preventing this early event in oncogenesis, but if you have a real tumor, it will do nothing to it. And therefore, we took authentic human pancreatic cancers, xenografts, growing in NSG mice, and we asked if we engineered these so that we could knock out the MUT1 gene at will, then what would happen to the growth of this tumor? The drivers of this cancer are G12D, P53 loss again, and CDKN2A mutations, and that's a typical combination in pancreatic cancers in people. And we found if we gave tamoxifen after the tumors were established and still fairly small, then a kind of growth arrest would occur, but they would regrow. We gave another dose of tamoxifen, and that knocked them down again. The problem is that tamoxifen isn't 100% effective at knocking out both alleles of the MUT1 gene. We had a little bit better result with A549 xenografts. This is a large human adenocarcinoma of the lung, and the drivers here are KRAS G12S and STK11, and this too was a lethal cancer in a human at some time in the past. Here, giving tamoxifen to the engineered cancer, where we knock out MUT1, causes real growth arrest, and for a very long period of time. This is what those tumors look like. All of them are much smaller than the controls where we didn't knock out MUT1 with tamoxifen. And you see the authentic size of these in cross-section here, but I'll blow up one of them for you. And I want to point out that the tumors still are there. You have live tumor cells, but they're simply not growing or expanding faster than they are dying or being eliminated. And that leads to the phenotype of non-growth. If you look for growth using Ki67 as a marker of dividing cells, you do see some dividing cells, though not nearly as many as you see in the wild-type tumor, where growth is mainly along the margins. And we know, as I told you, that there's a failure to hit every cell with tamoxifen. It's not a situation as though you had a real drug and there's constant presence of the drug and many opportunities over time for the drug to get into the cells and work its effect. The target protein is something, it's one of those examples where I have to be a little bit discreet because Potentium doesn't want me to disclose any targets at this point. But I will say it's not a very well-known protein, and it's nothing that anyone ever considered to be actionable for chemotherapy of cancer. We know that adult mice with a total body conditional knockout can live without the protein indefinitely, and they appear healthy, although they have some hematopoietic abnormalities. The germline knockout is lethal. We do have the original ENU allele, which is a viable, rather extreme hypomorphic allele, and those mice live as a stock, are fully fertile, and they are apparently healthy, and that makes us think that this is a very good target for chemotherapy. We did some other studies. In mice, unlike in humans, homozygosity for P53 knockout is survivable, and the mice develop something quite similar to human Li-Fraumeni syndrome, where they get sarcomas and leukemias and lymphomas, and that leads to their death. 100% of the colony is usually dead within 150 days or so of birth. And we looked at this situation too, using either homozygosity for the hypomorphic viable allele or the wild type form, and with homozygosity for the V allele of MUT1, there was prolonged growth, really perfectly healthy mice. Again, they maintained fertility. Again, you wouldn't be able to tell the difference between them and wild type. We did some other studies as well with DTAG alleles, where we could use a real drug that is off the shelf and is recruited to a new domain that we've added to the MUT1 protein, and in turn recruits an E3 ligase to drive degradation of that target. In this case, we used an eMumic B-cell leukemia, and it went instantly into remission with 4 doses of the DTAG drug. And this makes it really all but certain that a real drug would be useful. Finally, I want to say from our screening, we have a total of 4 mutations, MUT1 through MUT4, that have effects like this, and 2 of them work by one mechanism and 2 of them work by another mechanism. I show you here that if we combine floxed alleles of MUT1 and MUT2, and we do the same xenograft experiment with A549 xenografts in NOD-SCID mice, we get more than an additive effect. We can see that with MUT2 shown in the blue, we have a strong growth inhibition. With MUT1, which I've showed you before, we have growth inhibition. And if we have both genes floxed, we get even better inhibition. And this is how things stand now, but it may be that it goes on for the lifetime of the mouse. Notice that the tumor has shrunk from its original size, and we're really near a threshold where we can hardly measure the tumor. You can sort of feel there is something there, but we're using calipers, and I am not totally confident that it's not gone in some of the mice. So what to do about this? Obviously, this cries out for some kind of drug discovery effort, and we began by changing all of our methods in the lab and becoming computational chemists. My 3 computational people learned how to do docking, how to analyze with Boltz-2, an AI program for binding of drugs to targets, and how to use molecular dynamic simulation. And using these methods in that sequence, docking about 1 billion compounds in silico, taking the top several thousand and doing Boltz-2 analysis, and then taking the best 2,000 of those cases and doing molecular dynamic simulation, we came up with 50 top candidates, and 2 of them really did bind to our preferred pharmacophore, and we're working to optimize the binding now. We also did DEL screening, a wet lab method, and we found 2 binders that bind at other sites. In all these cases, we have solved the structure of the target protein, and we can see an extra density. We can fit the density to the compound, and from this point, we are proceeding forward with structure-guided drug discovery methods. We're at the hit-to-lead stage, and with our very best compound, we've reached mid-nanomolar affinity, and we have a very good site at which to add a recruitment domain. So I would say this is going very, very well, and it's something that I find really as exciting as anything I've ever done. So I'll just summarize for you what I think the import of this work is. Though I wouldn't have guessed it initially, there are many mutations that modify cancer and do so dramatically. Some act in a cancer cell intrinsic manner, some act extrinsically, and those mostly affect the immune system, and we can find either one depending what kind of screen we use. Now you might wonder how many there are, and we haven't gone in depth enough with our screening for me to give you a really clear answer, but I would be willing to bet there are certainly more than scores of them, because we've found almost 20 of them so far at a very low level of saturation. And I would say more likely there are hundreds of genes that could act this way, some probably quite broad in their specificity, others may be uniquely active against one kind of cancer or another. One might ask whether these loci that we're discovering influence human responses to cancers. You may have 2 patients, and they have identical oncogene drivers, and one does very well clinically and the other one does really poorly and dies very quickly. Could differences at some of these susceptibility loci or we could call them resistance loci, make that difference. And I think almost certainly yes, but in those things that we've found so far, I would say that on an evolutionary time scale, there would be selection against the mutations we're finding, even though they're quite tolerable in mice, and they would not exist at the level of polymorphisms, and they would therefore be very hard to find in human populations. Obviously, such mutations can point to actionable targets for drug discovery if they're well tolerated, and that's what we're doing now. And I would argue that this doesn't apply only to cancer, but to all kinds of diseases. If you want to understand how you might block degenerative neurological diseases, for example, expanding triplet diseases, metabolic diseases for which there are no good approaches, you could always do a suppressor screen in the mouse if the physiology matches that in the human fairly closely, and you could expect to find novel targets for drug discovery. So thank you very much for your attention, and if you have questions, I can try to answer them.
Thank you very much, doctor. Solo tenemos tiempo para una única pregunta, entonces, si alguien gusta.
Do you have an idea which proteins are these ones and in which pathways are they involved?
Yes, that's what I'm not able to tell you. We do know what the proteins are. We've solved their structure. We know a great deal about them, and we know a lot about the mechanism too. And I'll just go out on a limb and tell you that two of them are involved in one single pathway, and two others are involved in another pathway. And neither pathway has been explored really in cancer chemotherapy. None of them have anything to do with genotoxic effects. For example, we're not finding a new topoisomerase inhibitor. We're not finding a new alkylating agent. We're not finding anything that works on microtubules or is a spindle poison. Nothing that people have thought of before, which I think is really quite encouraging.
Thank you.
Thank you very much. So, sorry, Doctor, we
Okay.
have some time constraints, but on behalf of the Mexican Society of Immunology, I want to give you this. It's a little person from Mexico.
Well, thank
So
you
I hope
very much.
Thank you very much.